Hey everyone, welcome to our capstone pitch. We are Master of Engineering students from UC Berkeley. This project is supervised by Prof. Boris Rubinsky and Dr. Chenon Liu. We are presenting an innovative MIMS device to reduce the time and cost required for gene editing. New and revolutionary genetic medicines are transforming medical treatment. To better facilitate the development of precision medicine research, genome editing technologies are widely used. The CRISPR-Cas9 system is adapted into many genome editing protocols. This gene editing tool targets a specific DNA sequence and can precisely alter DNA by removing or correcting genes. Microinjection is considered as the gold standard for introducing the CRISPR system into the cell. Although this method is effective, it is a technically challenging and labor-intensive process because CRISPR has to be injected under a microscope using a needle with a diameter smaller than 5 micrometers. Alternatively, electroporation was introduced. The foreign particles are introduced into the cell through a small opening in the cell membrane created by an applied electrical field. However, the current electroporation method is time-consuming and expensive. Traditionally, electroporation is performed in bulk where a large voltage is applied to electroporate millions of cells at once. This approach is labor-intensive, time-consuming, and expensive, as it requires large quantities of the reagent and has limitations in efficiency of cell viability and transformation. A survey showed that a successful CRISPR experiment can cost about $18,000 for lab equipment and reagents, around 472 hands-on hours to perform the experiment, and 19 weeks to obtain a successful edit. Therefore, we investigated into a device which allows for controlled CRISPR delivery into a single cell using small amount of reagent. With the help of MIMS technology, a chip with a micrometer hole was fabricated. By applying voltage across the chip, electrical field concentrates at the hole area, which allows the hole to either attract or repel the particles through electrical forces. How we envision this device to work is first the cell will be placed in the liquid above the chip, which will be attracted to the hole by electrical force. Once the hole is blocked, we will increase the electrical force until electroporation achieved. Then the CRISPR will be introduced from the other side of the chip, and finally the cell will be repelled. Our goal for this project is to validate the possibility of using this chip to attract or repel the particles. This is the experimental setup we designed. The bottom chamber is filled with water and the top chamber is filled with bead suspension. Two conductive glasses are served as uniform electrode with either AC or DC power applied. The bead's movement is observed by a microscope from bottom. Here is the physical setup and an actual image of the beads we observed. Combining the COMSOL simulation results and the experimental results, we suspected that when the positive charge is applied at the top chamber, the fluid circulates down at the wall of the hole and flows back up from the center area, which brings the particle in the top chamber towards the hole and might repel it once arrived the hole. Oppositely, the circulation reversed when the voltage is reversed. We believe this technology is worth further investigation. Once developed, it could be a cheaper, less time-consuming, and more controlled alternative